Good afternoon, everyone, and welcome. So, step by step, this year's Open Readings Conference is coming to a close, and I hope you enjoyed it as much as I did, because for me, it seems as though this year's first invited speaker, Professor Luisa Torsi, was presenting only yesterday's morning, while in truth, we gathered here to listen to the last one. However, we're ending strong, and in just a few minutes, you will definitely learn something new and very interesting about the micro world below our feet. But first, I would like to introduce you to our invited speaker, Dr. Milda Pučetaitė. She is an alumni of Vilnius University, where in 2016, she successfully defended a thesis on vibrational spectroscopy and microspectroscopic imaging of urinary stones and biological fluids, and obtained her PhD in physics. Throughout her scientific career, Milda's work was focused on various applications on spectroscopy, such as fundamental studies of bonding of organic molecules, characterization of tissues for cancer diagnosis, searching for phylogenetic relations in fossil plants, or analyzing world-famous sunken Swedish ship. So, as you see, Milda has gained a lot of experience working on numerous projects, and also she has supervised a bunch of students, one of which I am honored to call myself as well. Well, maybe not officially, but still. Since 2018, Milda has been a postdoctoral researcher at Lund University in Sweden and is currently working there now. She's a member of the Soil Science Working Group and has started her own project where she will combine microfluidics and vibrational microspectroscopy in order to learn more about microscale fungal interactions. And today she will present us with some of her findings and will show us how advanced spectroscopy methods can shine a new light into the soil systems and help us tackle environmental problems. So let us all welcome her with a round of virtual applause. And I pass the microphone to you, Milda.
Thank you, Martini, for a very full introduction and very nice introduction. Let me share my screen first. Good afternoon to everyone again, and thank you again for introducing me. Thank you also for the organizers for inviting me to speak. I'm actually very nervous to follow all the other invited speakers who have been presenting this week, but I hope that you will be interested in hearing about the work that I have been doing in the last few years in Lund University, where I have been working with applications of spectroscopy techniques to soil systems with the aim to tackle related environmental challenges. I prepared a slide to introduce myself a little bit, but Martina did it really well for me, so I will not spend a lot of time here. But just to reiterate, I'm a graduate from Vilnius University, where I have worked with applying spectroscopy techniques to many different biomedical samples with the aim to find diagnostic and disease prevention techniques. Then I moved on to Lund University as a postdoc, where I added to my spectroscopy and microspectroscopy toolbox a new technique, which is synchrotron radiation-based Xray microscopy. And here I discovered the unknown new world of soils, since I have been analyzing soil processes and soil aggregation, which I will be telling you more about today. Liking this topic quite a lot, I will now continue as an independent researcher in the university, and will continue applying different types of spectroscopy techniques to soil microbial life and processes and analyzing processes that they govern. So why did I get interested in soil after studying biomedical samples for so long? For one, when I started the project that I am in now, I came in there purely for learning a new technique, but then I discovered that soil is not at all what I thought. It's not just mud basically under our feet. It performs a lot of vital functions in our lives. Maybe the most obvious one that we rarely think about is providing us with food, fibers, and fuels through agricultural practices. Another is that soils are one of the most biodiverse ecosystems in the world, with soils housing many different organisms, from tiny bacteria to larger insects and animals. And maybe one that is less known is soils are sinks of carbon and regulators of climate. And this is the function of soil that I have been particularly interested in. It seems that more and more governments and entrepreneurs are interested in finding biotechnological tools to sequester carbon from the atmosphere, which we humans emit excessively into the atmosphere. However, the largest sink of carbon is actually just right under our feet. So let me explain a bit how and why carbon stays in soil. This is a rough scheme of carbon cycling, and carbon in the cycle, carbon in the atmosphere is getting absorbed by vegetation on Earth, and through photosynthesis converted from carbon dioxide to organic molecules. These organic molecules reach soil by fallen vegetation like leaves and roots, where they are subjected to microbial decomposition. I call it a kind of a recycling factory, where all these organic molecules are recycled. And then the outcome of this recycling is that CO2 is emitted right back into the atmosphere. So in order to answer the question of how to store carbon in soils, we really need to understand why is there carbon in soils overall? Why is there more carbon in soil than in the atmosphere and the biosphere combined? After all, in a handful of soils, there are more microbes than stars in a galaxy. And all of these microbes are hungry for food. They are hungry for these falling organic materials, and they want to eat it. So what prevents them from eating it all? Before, I have shown soils to you as just brown pile of mud, but in reality, soils look much more like this. It's a complex network of minerals, water, and air, where the microbes and insects and animals live. And if you're yourself a microbe, then you see soil much more like this. This structure, which you see a video of now, is a simulation prepared using structures of soil obtained through X-ray microtomography. And you see how intricate it is. And when microbes navigate the soil, it's really like a Pac-Man game for them. So where they have to go through these tunnels of minerals in order to find organic matter that they will use for food. So really, the soil structure is the answer that we are looking for. And the soil structure is hiding organic matter from the hungry microbes. So in order to find out how to store more carbon in soils, and how to maybe make soils account for at least part of the excessive emissions that we emit into the atmosphere of carbon, we need to find ways of how to use this soil structure to put more carbon there. And for this, soil scientists are studying soil, but it's not an easy object to study, really, because soils are completely opaque to visible light. We cannot also see the tiny microbial life living there. nor we can see the organic matter that falls there, apart from the layer that is on top with the falling leaves. So advanced spectroscopy techniques can come to aid in answering these questions and getting insights into what life is really like under our feet. I'm sure that the word spectroscopy is not new to most of the audience here, but just briefly, it's a study of light-matter interactions as a function of wavelength. Depending on the wavelength that is used, we can get a different type of information about the materials using light. For example, if we use vibrational spectroscopy, which is using visible or infrared light, due to interactions of this light with the material, molecules in the material, we can get information about molecular composition of samples. On the other hand, if we use X-ray spectroscopy, due to interactions of X-rays with the inner shell electrons and specificity of electron levels for each element, X-ray spectroscopy gives us information about elemental composition of samples. Furthermore, spectroscopy can be performed in bulk samples, where basically we take a bulk sample, for example, in this case, soil. We shine light into it from a radiation source, and then record a spectrum, which is basically a result of the light-matter interaction. And the peaks in a spectrum are basically resulting typically from some sort of absorption or specific scattering phenomena in a sample, which then allow us to analyze those spectra and relate this to the chemical composition of a sample. Also, furthermore, spectroscopy can be used in microscopy mode, so-called microspectroscopy. And here, the radiation from a light source is focused to a small point on a sample, the point diameter being limited by the optics that we're using to focus it, and of course, the diffraction limit. And this allows us to not only look at the chemical composition of a sample, but also by scanning the sample in the path of the focused light, we can figure out the distribution of different components within, which can be very useful when we are analyzing heterogeneous samples. From the beginning of this talk, I have been using a term advanced spectroscopy, which here I mean that typical and more custom spectroscopy approaches are improved by state-of-the-art light sources, or, for example, detection methods to increase spatial, temporal, or chemical resolution. And in my work, more specifically, I have been using advanced spectroscopy as supported by synchrotron sources as sources for radiation to perform spectroscopy. In a synchrotron, there's a huge ring where electrons are accelerated and bent, and the result of this acceleration and bending is emission of electromagnetic radiation, which is then extracted to so-called beam lines, where spectrometers can be placed and spectroscopy performed. Synchrotron radiation source features high brilliance, which improves sensitivity of our spectroscopy method. Also, it entails that the radiation can be focused to smaller spots, especially for X-ray. This is very relevant since while focusing X-ray radiation, a lot of light is lost in the process due to the optics required for focusing. So we can improve lateral resolution using synchrotron and the high brilliance of the synchrotron light. And finally, it also features a broad spectrum, which means that we can perform different types of spectroscopy, from infrared to X-ray spectroscopy, and then complement information that we have about the sample. Synchrotron source is not easily accessible and not easy to have in the lab. It's impossible to have in the lab, of course, but there are typically state-owned synchrotron sources around the world where one can apply for access and to perform their research. It's quite a competitive process to apply for this access, but it's definitely worth it to gain state-of-the-art knowledge about the samples. And then in my research, I have been using several synchrotron sources. One of them, MAX4, is conveniently just here in Lund, next to university I work in. But I have also traveled to a synchrotron Diamond in the UK, Soleil in France, and a Canadian light source in Canada. And this traveling is required in order to be able to get more replicates of your sample and perform more experiments, just because it's difficult to get enough experiment time in a single facility. So what does spectroscopy of bulk soils give us? We can take a handful of soil that I mentioned before, where all the microbes live and the organic matter hide, and perform different types of spectroscopy techniques on this bulk soil. We can, of course, obtain very useful information from this. We can get elemental composition of soil minerals. We can obtain molecular information about organic and inorganic soil components, which could allow us to predict the total carbon content in soil, for example. However, if we really want to understand what is happening behind this structure of soil and how to make organic matter to hide in a structure of soil, we really need more information about what processes are behind this organic matter hiding in between the minerals, and how these processes can be further employed for carbon storage in soil. And when we take bulk soil, what we basically do is just destroy its structure. When the structure is destroyed, we cannot predict much about what made it in the first place. So then in my research, I have been using mostly microspectroscopy tools, which allow me to really look more at soil structures and more directly at soil microbes and how these soil structures and microbes and organic matter interact all together, and what processes take place there. I have been doing this in several different projects, and the first one I will talk about is the one where we have been investigating soil aggregates. Soil aggregates are basic units of soil structure, basically. They look something like this when you look at them in a field, in soil. And basically, this one piece of soil, this one soil aggregate, is this labyrinth that I keep showing you, where organic matter hides. So in order to find out how we can hide organic matter in these structures, we performed an artificial soil aggregation experiment. We basically took completely mineral soil, deconstructed it into basic mineral particles where no aggregates exist, and then re-aggregated it in the lab by using selected organic matter sources, in this case plant litter from straw and maize. These two litters from plants, they feature very different physicochemical properties and also nutritional values for the microbes. So we expect that these two litter types will be a good model of what type of properties would cause the organic matter to hide or attach more to the minerals, which would then hide it from the microbes, and which organic matter maybe the microbes would go for more to then decompose it. We have used a synchrotron-based Xray fluorescence microscopy to analyze our soil microbes sectioned into thin sections, and to be able to analyze the traces of this litter in there, we needed to label them with rare earth labels, neodymium and samarium. Let me explain why we needed this labeling. X-ray fluorescence microscopy is a technique which gives us information about elemental composition of samples. The fluorescence is excited when an incident radiation from a light source, in this case a synchrotron source, interacts with the inner layers of electrons in an atom, and then ejects them into unoccupied electronic states or a continuum. Then a formed core hole is created, and later is filled with electrons decaying from top layers of electron shells, electron levels, through radiative path and emitting fluorescence photons. Because every element has a specific electronic structure, the fluorescence is specific to different chemical elements. So, for example, in a spectrum, we can differentiate aluminum, silicon, potassium, calcium, manganese, or iron. But for lighter atoms, such as, for example, carbon, the decay typically is much less probable that the decay here will be radiative, also, which then reduces the intensity of the fluorescent signal. Also, the fluorescence of the carbon atoms is in a spectral range, which is easily absorbed by air, which means that if we have air-filled microscope, which due to technical limitations usually is, by the time the radiation travels from a sample to the X-ray fluorescence detector, there's no more fluorescence from a carbon source. So then in order to look at carbon, we need to label it. We use the samarium and neodymium because these two elements are rare elements in soil, so they would not interfere with any other signals. And in general, X-ray fluorescence, it was useful to be able to see all the elements of soil, and then through labeling organic matter in our aggregates. What we did is put our aggregate section in a sample chamber and then scan it in the path of X-ray beam, and then obtain these type of chemical images of the elements within. Here we can see a distribution of different elements that are mostly present in soil minerals. And then through samarium and neodymium, we can see distribution of our maize and straw in this aggregate. What we found is that in small structures of soil, in micro aggregates that are smaller than 50 microns, it doesn't seem that there is any type of specific distribution of different types of litter source of maize or straw. But when we look at the bigger structures of soil at macro aggregates that are larger than 250 microns, for example, then we can clearly see that for maize, which is in red here, form big clumps of organic matter, basically within the structure of the aggregate. Here I show only the image of maize and samarium, maize and straw, because of the limitation of space. But we also can see from other images that these clumps of organic matter are surrounded by minerals, which means that for the microbes, it will be very difficult to access these maize clumps, and it will not be decomposed or decomposed with smaller probability. So what does this tell us? It tells us that different vegetation types in soil can have a different influence on soil structure and subsequently carbon storage. And this is important to know because climate change changes the landscape a lot. The vegetation in the landscape changes, and that means that the carbon storage capacity in certain soils will be altered. At the same time, it's important to know for farmers, for example, if they want to increase the carbon in their field, the carbon content in their field, and depending on what soil is in their field, they can use, for example, maize. They know that that maize will probably stay in soil with high probability. Yeah, that brings me to the next project that I have been working on, which then goes even deeper into soil. We're going from the aggregate scale to a single mineral particle scale and see how these mineral particles interact with microbes in soil, and more specifically fungi in soil. And let me explain why this is interesting. It seems that fungi, compared to, for example, bacteria, play a particularly important role in regulating soil processes. When you think of fungi, you might think of maybe mushrooms that you pick in the forest for food, or if you don't like mushrooms, maybe you can think of beer, because the fungi are also responsible for producing your next pint of beer. However, in soils, fungi most often look like this. Their majority of biomass of fungal biomass is underground, where they live in symbiosis with the plant and plant roots. And this symbiosis entails that a plant produces carbon and then sends it to the fungus to feed it, while fungus extends deep into the mineral soil to look for mineral nutrients, which it then supplies the plant with. This extension of the fungal cells, which are like this elongated compared, for example, to typically round bacterial cells, means that soil fungi are the main vectors of organic compounds that they get from a plant into mineral soils. And with our project, we wanted to figure out how this happens. This had been predicted that this happens, but we wanted to know of what process is responsible for fungi delivering organic compounds into soil. So the experiment that we performed was what we call fungal mineral interaction experiment. We took several different species of fungi with different nutrient foraging properties or strategies, and then have grown them in contact with two different types of minerals: quartz, shown here on top, and goethite, iron-based mineral here in the bottom. We chose this experimental scheme because we wanted to see how the fungi interact with a different type of minerals. For example, quartz is a silicon oxide where the fungi can barely find any nutrients. However, goethite contains a lot of iron, which is essential for functioning of fungal cells. So we predict that fungi will be going after this iron in the goethite and will deposit likely more carbon on the goethite. The way we performed this experiment was in the lab, not with the mushrooms that I'm showing you here for visual impact. We have performed experiments in Petri dish where we have grown the fungi, and then just in front of the fungal cells growing in these Petri dishes, we placed our minerals, which were dusted on Xray transparent optical windows, which as a result then look like this. This is an optical window, the fungal cells growing, and then on some of these windows there are mineral particles deposited. And then for analysis of these samples, we have used X-ray absorption spectroscopy that provides information about, again, elemental composition of samples, but different than fluorescence spectroscopy. It provides also information about bonding and speciation of elements. In X-ray absorption spectroscopy, absorption is created by interaction of X-ray light with again inner shell electrons, which then are expelled when from the electron level when the X-ray photon is absorbed. Then scanning through different energies, we can see different absorption edges for different elements. It's so far similar what we saw in X-ray fluorescence experiment, only the shape of the spectrum is different. However, if we zoom in into this absorption edge of a single element. For example, let's look at carbon. We can see that this absorption edge has a structure, and this structure is determined by the bonding of the element that we're looking at in a molecule in our sample, which means that we can look basically at and predict, at least in a broad sense, a molecular composition of our samples. Again, also different from the fluorescence experiment, that these type of microscopes, X-ray absorption microscopes, are in vacuum, which means that now we can look at the carbon compounds by using suitable energy ranges, and the air is not interfering with our spectroscopy type. However, the minerals are more difficult to see. This is why we could not use this technique for the aggregates, because in the minerals, this radiation would be, radiation in this energy range would be highly absorbed. The X-ray transmission microscopy experiment is again performed by placing a sample in front of the focused X-ray beam and then scanning it to obtain map of an area of interest, and then scanning through different X-ray energies to collect a full spectrum of X-ray absorption. And then these spectra are analyzed using multivariate principal component analysis and cluster analysis to be able to differentiate where different chemical compounds are located in our samples. So this is the result that we obtain for a fungus, which is called Psilocybe subviscida, which is a relative of the magic mushroom that maybe people are familiar with. We have a cell growing in a contact with a quartz mineral particle. And then here in an X-ray transmission image, we see that the quartz has a very low transmission. Most radiation here is absorbed. And the higher transmission is observed in areas where the cell is located, in the areas where the organic fungal cell is located. Then in the chemical image, different colors show us different chemical compounds in the cell mineral system. The green here shows proteinaceous hyphal cells or fungal cell. In blue, it's the mostly chitin-based fungal cell wall. In white is again the quartz mineral particle that here we also see the spectrum of it. It absorbs X-ray radiation to a great extent, and then the spectrum is featuring a lot of light saturation. And then in red, just around the mineral particle, we have found that the fungus have deposited some slime, which is sticking to the mineral surface, and we can see that this red slime is even on the other side of the particle, which is probably due to some wetting effects. The particles that we have put in the system were clean, so we are sure that this slime is coming from the fungus. We have observed this also for another species of fungus in contact with, for example, goethite particles. Here they are also in gray in this needle-like shape. And this slime is different for different species we found. For the fungus here on the left, for the Psilocybe, we found that the slime is mostly sugar-based, while the slime around the goethite exuded by this fungus, which is called Bacillus involutus, it's mostly protein-based. This could be due to the fact that the different species of fungi are exuding different types of materials as the slime, or it could be due to the fact that the slime absorbs differently on different minerals. This we need to look further into, and we are still performing more experiments now on this. What we did do, though, is to be able to see if this protein-based slime on the goethite particles interact with the mineral. We have analyzed this particular sample, not only in the energy ranges where the carbon is absorbing, but also in the energy ranges where iron is absorbing. This is how an iron absorption edge looks like. And again, this spectral feature is specific to a mineral goethite, since X-ray absorption spectra give us information about the bonding of iron, but also the two peaks are characteristic to different speciation of iron atoms, iron 3 and iron 2. Goethite is mostly iron 3 containing mineral. And what we saw in the mineral particles that were in contact with the fungus, that the intensity of the iron 2 related spectral band is increasing, which means that the fungus is reducing the iron in the mineral. Reduction entails that the solubility of the iron from the mineral is increased, which means that the fungus is probably trying to extract this nutrient from the mineral and uptake it to its cell. So this is quite an interesting discovery, which has not been observed on a single cell level. What does this whole experiment tell us in general? It tells us that while mining for inorganic nutrients from minerals, fungi deposit large amounts of carbon compounds on them. And there, in a depth of soil, this carbon is hardly accessible to other decomposers such as bacteria, which mostly stay on top of the soil where litter sources are available. And it is important to know this because with our agricultural practices, we destroy a lot of fungi, which means that we probably prevent a lot of carbon to be deposited into soils. So for example, with the deforestation, I have told you before that the fungi mostly are living in symbiosis with trees, with plants. And then if we cut the trees, of course, then the fungi cannot survive either. In also fields where tilling is performed, the hyphal structure of the fungi is destroyed and they also cannot survive. So in performing these agricultural practices, we really need to think about how much fungi we will destroy and how that will affect the capacity of carbon storage. So far, I have been talking a lot about these experiments that are really fundamental. I have been looking in these artificial laboratory produced experiments. And the but here is how much does this really matter in soil? It's difficult to say. From the beginning, I was telling you that soils are difficult to study, and this is true. It's impossible to just do spectroscopy in real soil from real environmental system. So scientists are looking more and more for approaches that would allow us to bring more realistic soil systems into the lab. And how can this be done? Very important piece of technology that has been recently applied in soil science is microfluidics. Historically, microfluidics has been used for fluid mixing and following up on chemical reactions. But in soil science, people in my group specifically have been working a lot on adapting microfluidic technology to mimic soil structures to make it more realistic, the experiments in the lab. So what we do is create this type of rubber and glass based microfluidic chips where microstructures simulating soil structure are engraved. Then we bury these microfluidic chips directly into a forest, for example, soil. And after a while, once these microfluidic chips have been colonized with microbes and and fungi and bacteria and other organisms, we can excavate them and then look at them under the microscope. And this way, microfluidics, or what we call soil chips, literally serves as a window to the underground. So we have real soil, real microbes, but in a transparent microfluidic chip. Here's just some example of an experiment that has been performed in our group, where a microfluidic chips with soil imitating structures have been used and buried in the soil. These structures are not strictly imitating real soil. However, they are producing the structure that is possible to replicate and then quantify certain processes in. And once these structures are colonized by soil organisms and particles, we can observe a lot of different processes. We can see how mineral particles move around in these microfluidic chips, how all of the structure made by these mineral particles is affected by water flows. We can see how fungal hyphae move around in different structures. And then we can also see how these fungi interact with mineral particles on a larger scale, not only on with a single particle, also in a more realistic setting. We can also see a soil food web where different organisms eat each other, and that also contributes to organic matter turnover in soils. Here, I also have a small video to see how all of this life in soil move around. So small nematodes are trapped in a structure of fungal hyphae here. There are some small microorganisms moving around, and then even tiny bacteria are living in this soil-like chip, and then that we can observe them under a microscope. So then the next step that we want to work with is bring these microfluidic chips to a spectroscopy or microspectroscopy device and see whether we could if we could tell something more about what chemical process is happening within, and whether the chemical process is taking place in a chip where the soil structure is kind of simulated differ compared to the experiments that we have been performing on very simplistic laboratory systems that I have been showing you before. And this is what my future project is related, is using vibrational, infrared and Raman microspectroscopy in these soil chips. As an overview, I have this slide where it's kind of sketch of how this could be done, how a microfluidic chip could be put in a pathway of different types of radiation, for example, X-rays, infrared or lasers, and then how the response from within the chips could be collected to, for example, look again at the mineral interactions with organic matter, for identifying microbes or microbial slime, or even heavy metals in soil as pollutants, potential pollutants. Of course, this would require also certain changes in the microfluidic chip design to make them transparent to the type of radiation that would be using for spectroscopy. And to elaborate more on the project that I will be working for the next four years, I want to answer the question about how chemistry of the fungal interactions affect carbon emissions from soil, or again, carbon sequestration in soil, if we look at it from the other way around. I think throughout my talk, I already expressed my admiration of fungi that I kind of gained through the years of working with them. Also, I said that fungi are pretty abundant in the soil, and where there's abundance, there's also a competition for nutrients. And this type of fungal warfare, what we call fungal warfare, affects the spread of fungi and their growth and the composition of organic matter. And then, as a consequence, they also that also affects potential carbon sequestration and carbon emissions from soil. And for this type of fungal warfare, I want to use the microfluidic chips that would allow me to look at the fungi in a semi-realistic environment, would also provide the possibility to separate individual cells for analysis and look at the processes that right at the forefront of or hotspots where they appear, so at a single cell scale. The chips would also allow me to simulate the chemical environment with high precision and, of course, provide optical access to the system for the spectroscopy techniques. I have several techniques that I have in mind in using. One of them is Raman scattering microspectroscopy just in our lab, and the other one is a new technique. I will not go much into detail with it, but it's a new technique that allows to look at the molecular composition of samples with infrared microspectroscopy-like approach, which allows us to break down the diffraction limit of infrared microspectroscopy, custom infrared microspectroscopy, which means that we can really look at the high resolution, highly resolved processes happening at the fungal cells with this technique. And as a summary now, I was speaking, I think, quite enthusiastically about how much spectroscopy can do in soil science. However, I don't think that spectroscopy in general can solve all the problems in soil science or answer all the questions. But it definitely brings us closer to understanding nature and its responses to environmental change, which is, I think, important to study. And I'm happy to be working with and bring this knowledge of the techniques to a very interdisciplinary field of soil science. And I think as a last slide, I want to say what for me is the most important thing in research. It's not the techniques, it's not the research question. I think it's the team that you're working with. And especially if you're working in a very interdisciplinary projects such as mine, I think it's very important to communicate with scientists from, in my case, from biology, from physics, from engineers of microfluidic chips, and so on and so on. Because without them, it would be very difficult to perform this type of projects. I'm definitely not an expert of soils, but I'm happy to have people who guide me in this world of soils, and I'm happy to contribute to the project with spectroscopy knowledge. So yes, thank you for listening, and I can ask questions.
Yes, thank you very much, Milda. That was a very nice and really informative presentation. So we have time for all of your questions. So do not hesitate to ask yourself. You can also write it in the chat, or if anyone is watching on YouTube, write it in the comments and we will read it out to Milda. So I think everyone is still processing the information they just received. So
a lot of biology.
Yeah, and so before everyone else, I then would like to ask you, I think you mentioned this already in a couple of last slides, but so my question was whether these spectroscopy methods could show us all the information that we need, for example, to tackle the climate change in regard to soil?
I don't think it can answer all the questions. I think the important point, which I don't know if I managed to highlight, is that combination of different techniques, as well as combination of different scientists from different fields, is very important to tackle such complex problems that climate change and environmental change in general poses to us. So soil, as well as other systems, I think in biology, the same, I think I experienced also working with biomedical samples. They are really complex, all of these problems that they pose, and just one technique or one person, I think, would not be able to solve them. However, I think it's important to interdisciplinary collaborations to bring us closer to answering certain questions about what happens in these systems, what processes govern interactions with the environment, with what governs interactions of microbes, how they govern then the carbon cycle, and so on and so on. So we can answer certain questions with different types of spectroscopy techniques, also with different types of other methods, and then combining this information together. That is what, and then of course, transferring these answers to some kind of practical applications. I've been mentioning some agricultural practices, which I don't know much about, but I kind of envision that if we have some knowledge about the processes that happen, we can make a recommendation for practical applications that then would have implications for climate, for example.
Yeah, and also maybe a question from the drawbacks of spectroscopy. So regarding these microfluidic chips, how is water influence on the infrared absorption spectra? Is the sample so thin that it doesn't matter? I think it still should matter, at least in some way.
So yes, water does matter, and the main tool that I will be using this is Raman microspectroscopy, where water is less of a problem, especially since the volume of water is so small. Also, I think I mentioned, I didn't elaborate on it broadly, but I mentioned that I will not be using the typical infrared absorption spectroscopy. I will be using this diffraction unlimited infrared spectroscopy, where the detection is actually of compounds is indirect. So we basically excite infrared or vibrations in the molecules in a sample with the infrared laser source, and then we detect these vibrations and the photothermal effect that they cause, so the expansion of the material that the vibrations cause, by using visible radiation and the changes in the reflection of a visible laser. And this type of indirect detection of the vibrations in a sample, at least it has been shown in literature, it's reducing the effects of water or influence of water in the spectra. So I'm very curious to try. I actually have to admit that I have not tried this particular approach yet, but I'm hoping that it will be possible.
So this is some kind of nonlinear, right? Or still a linear spectroscopy? Because other question of mine would be, what about nonlinear spectroscopy, like cars or something like that?
The photothermal infrared spectroscopy is a linear process. The vibrations are excited just like in infrared spectroscopy, just using the laser source. And then what is tracked is basically the expansion of material that is caused by the, so it's kind of optical mechanical detection. So it's a linear process. Cars, which yes, would be an nonlinear spectroscopy. I guess it could be used as well. I mainly did not consider it so far because we don't have this system here. But yes, I think I would be curious to see whether it would be possible to do it. There has been a lot of interesting research on laboratory systems of soil with cars, and I wonder if that would also work in microfluidic chip. That would be interesting to try. Yes. Okay.
And also, I know that well, at least in Lithuania, one of the biggest issues of this carbon release from the soil is, of course, agriculture and all the stuff that would put in, like fertilizers, pesticides. So do you know, are there any research has been done on the fertilizer impact on soil? Like we see that fungi can extract elements. What about these other chemicals, for example?
A lot of research is being done on that. I'm by no means an expert on this, but a lot of research is being done on that, and it seems that using fertilizers on soils actually reduces the amount of different microbial life in soil. it's because it seems that the plants then doesn't need the microbes so much. So plants kind of use the microbes to obtain mineral nutrients, but once they have the fertilizer, they don't need it. And then this microbial life is kind of diminished in these type of soils. And then diminished microbial life means that the processes that that take place, that are regulated by the microbes to sequester carbon, are not taking place. So they do have, so these fertilizers do have a lot of effect, not to mention that they also pollute the environment through runoff to the groundwaters and so on and so on. So yes, a lot of research is being done on that, and that influences ecosystems in a severe way.
Yeah,
and yeah, spectroscopy, I guess, could also be used for that, mainly to track how these fertilizers, especially organic fertilizers, how they move around in soil, where they end up, and how they influence the microbial life.
Yeah, thank you. And maybe the last question from me. I hope somebody will have other questions, but there are very much types and varieties of soil. So how can we put that information into one, I don't know, catalog? What should we do? I mean, because one soil type may be acting in one way, other in a different way.
Yes, yes, of course, that mineral properties is like a very important parameter for any processes that take place, as well as where that soil is located. So how much moisture, what is the temperature is in soil, and so on and so on. So yes, once the research is done on certain soils, it's cataloged, or I don't know, recorded also in a scientific literature, of course. And then to generalize all of this information is quite difficult because the soil is so diverse.
Yeah.
It's true. But at the same time, if you could obtain information for one soil type, you could already give certain recommendations for managing that one soil type, which is already something. And then you move on from there.
Yeah. Okay, thanks. So I would like to ask the audience, do you have any questions now?
Yeah, I would like to ask, is there a reasonable explanation why soil tiling should be used, or it should be banned, or how it should be done?
So again, I'm not a big expert on agriculture, but I think that more and more tilling or soil tilling is being discouraged in soil agricultural practices, just for the fact that tilling really destroys the soil structure, which I have mentioned throughout my talk, that is so important for keeping carbon and keeping microbes alive there. And then having carbon in soil and microbes in soil then also increases the fertility of soil. So then you would need less fertilizer. So overall, if we would kind of reduce the certain tilling practices, probably we could in time regain natural fertility of soils. Of course, I guess there are some other factors to consider that I might not know about. But yes, I think that tilling is typically discouraged more and more from being used.
Thank you.
About other questions? Well, I don't see. So, okay, we don't have any questions left, I guess. So if you have any questions later on, I think you can definitely write to Milda. She would happily answer your questions. So once again, thank you very much, Milda, for your great presentation. And I guess we'll see everyone in the poster session now. So thank you very much.
Thank you. Have a nice end of the conference.